present on intelligently designed ontology alignment and a case study from the sequence ontology.
Okay, hello. My name is Michael Sinclair. I'm a postdoc in the lab of Karen Eelbeck at the University of Utah. I'm working in collaboration with Mike Beta from the University of Colorado, who works in the lab of Larry Hunter, who was here yesterday, but I don't see him today. So Karen Eelbeck maintains the sequence ontology, or SO for short. And for a while, she's been working on a companion ontology called the molecular sequence ontology, or MSO for short. And my task is to ensure that these two ontologies, which are tightly linked conceptually, evolve from the outset in parallel with each other. And this is what I mean by alignment here. So I do not mean the field of alignment where two already existing ontologies are compared to see how closely similar they are semantically and structurally. Rather, we aim to make these two ontologies evolve from the outset to be maximally aligned. So that, in this presentation, I will describe how I do that in the hope that perhaps other people may find it useful for their own problems in designing two companion ontologies and keeping them aligned. And also, and importantly, I want to inform users of the sequence ontology who are used to the way it has been, and potential users of the MSO, so that they understand the changes that are occurring and how the SO is going to be changed, and how the MSO could actually solve problems that they may be having in forming cross products and logical definitions between the SO and other ontologies, neighboring ontologies. So by way of background, SO is a formal representation of features of interest in macromolecular sequences of particularly functional and structural regions, such as introns, exons, genes, poly-A tails, types of variations like single nucleotide polymorphisms, and sequence assemblies like genome assemblies. It's important to understand that the SO terms describe features of sequence that are completely abstracted from molecular context. So you've got abstract entities like gene models when you pull up in like the USCS genome browser and you get gene models. These aren't actually molecules themselves, which can exist in any number of copies in all the different chromosomes in the cell of an organism, or maybe they're predicted genes that have not actually been found to exist in any single cellular organism in any species. So I just want to stress the abstract character of these entities. All ontologies need a kind of philosophical underpinning as to how they categorize entities in reality, and the SO is no different. Now, the SO, as with most open biomedical ontologies, aspires, at least in name, to adopt the basic formal ontology, or BUFO. And however, it doesn't really, and that's part of the problem that we are trying to address. And I don't have time to explain all of the ins and outs of BUFO, but I hope those who aren't familiar with it can follow along, because I only use a small part of it in this presentation. So the sequence ontology terms as information entities, they are what BUFO calls generically dependent continuants. Okay, so these are these are information entities like the text of a book or the sequence of a gene. They can exist as multiple copies in multiple bearers. So the bearers of types of sequence are biopolymers like DNA and polypeptides. And these molecules are physical entities distinct from the information content of their sequence. So there's a need to distinguish between the sequence information and the molecules that bear them as physical entities, because some ontologies need to make logical definitions that involve sequences as molecules rather than information, and as, for example, to say that a spliceosome splices an intron. That's a chemical reaction that it operates on molecules and not on, say, introns as information entities. And so while the current SO insists that its entities are generically dependent continuants, they actually, for the most part, write definitions that more or less treat the terms as physical molecules. So aside from some very general terms for molecules, I lost my cursor, for general types of molecules as found in CHEBI, chemical entities of biological interest, there isn't a detailed ontology of sequence molecules as molecules. So to solve that problem, we are launching the molecular sequence ontology. And this is the MSO for short, has been under development for a few years before I came on the scene. It's been development by Mike Beta and Karen Eelbeck. And it is to be a formal representation of macromolecular entities, molecules, whose order of monomers carries sequence of biological significance. So for example, DNA molecules, RNA molecules, and proteins. And the molecules are what Buffo calls independent continuants, and that's a fancy name of saying that they're a thing, a physical thing. That the important property of independent continuants is that they are the bearers of generically dependent continuants, such as, for example, when a physical book bears a sequence of letters that forms a linguistically meaningful text, which is on its own a type of information that's independent from the actual book in which it is written. Or for when a DNA molecule bears a sequence of nucleotides that forms biologically meaningful instructions to a cell that in a sense is independent from the actual DNA molecule that encodes it. So MSO entities as independent continuants are bearers of the sequence ontology entities, which are informational annotations about features of sequence. This relation, or the property that's going to be used in the web ontology language, or OWL, and from now on I'll just be assuming familiarity with the terminology of OWL, linking the MSO with the SO is the generically depends on relation, a relation that's defined by the Buffo standard. So a sequence ontology entity generically depends on an MSO entity, and the inverse of that is that an MSO entity is a generic bearer of an SO entity. So by representing MSO entities as molecules, there we go, sorry, as molecules that can engage in chemical and physical reactions, they can be part of logical definitions from neighboring ontologies that require independent continuants, molecules as participants. And so here's a very illustrative example of why the MSO is needed. It fills a very real need. Now, this is an actual issue raised on a gene ontology GitHub repository here. So a subset of CHEBI was being reused in an ontology involving Go terms, and the logical unsatisfiability arose because of this confusion between information entities and physical entities. So CHEBI messenger RNA was said to be equal to the SO mRNA. They thought that this was just the two terms for the same thing. But this axiom was unsatisfiable because CHEBI is a subclass of independent continuant. CHEBI strictly follows the Buffo scheme of categorizing entities in the world. So CHEBI entities are independent continuants. But it was unsatisfiable because the SO mRNA is officially generically dependent continuants, so they cannot be equated. They're subclasses of two totally different entities. Likewise, SO transcript, it cannot be a subclass of CHEBI ribonucleic acid because for the exact same reasons. So the correct axioms that aren't in place yet that would be enabled by the new MSO ontology is one setting the CHEBI mRNA equivalent to molecular SO mRNA, a class to be introduced in the new ontology, and then specifying the SO mRNA for convenience using the same names for now, generically depending on the MSO mRNA in order to connect the two via the MSO. And this last axiom is the key for our purposes, and I want to impress it upon you and have you keep it in your mind for the rest of the presentation. It's very important here. But the upshot of the molecular sequence ontology is that by creating it and making this logical connection via the generic dependence with the MSO depending on the MSO, we bring the SO out of its silo. It was very siloed in terms of a data source. It could not interact with Go or any other molecular-based ontologies. Now we can relate an mRNA annotation in a database with a gene model to an actual mRNA chemical through molecular sequence ontology, and it can now participate in cross products with the gene ontology and other ontologies, and they've all conformed to the basic worldview of the basic formal ontology, which imposes a logical structure that allows cross products to be built with a lot more consistency and satisfiability. So that was a lot of background here, but my goal, what am I doing? I'm basically a OWL API software developer here, and so I'm trying to make sure that the SO and the MSO stay parallel by automating the process of adding new terms, so that whenever the MSO is updated, or vice versa, the SO is updated with a new term that somebody requests, that they are updated in tandem and vice versa, so that the curator does not have to manually update both ontologies separately. And in so doing, we reduce the risk of human error and we save time. You know, the motivations for a lot of our automated strategies here. So our strategy for this is to employ a single master file. This master file contains all the terms for both ontologies, and it's the sole file that the curator needs to update manually. From this file, we will automatically generate the separate MSO and SO ontology files using a custom program written in the OWL API. Everything I've done towards, so writing this program here in the OWL API. Now, we need to choose one of these two ontologies to really be the primary one for the purposes of adding our terms and basing one on the other. The choice is not crucially important, depends more or less on one's philosophical orientation. We've chosen the MSO to be the primary ontology here, with the understanding that each MSO term, or most of them, will have a counterpart SO term that generically depends on it. So when the curator adds terms to the master file, he or she would add them as SO terms and write the definition as SO terms, with the understanding you're going to have a counterpart SO term that generically depends on it. However, there will be cases where some terms in the SO will not generically depend on any MSO term, and likewise, you will have some MSO terms that won't relate or generically bear any MSO terms. So for example, a read, SO read, how can this relate to MSO? It's a DNA sequencer's best guess as to the actual sequence of an arbitrary fragment of DNA, the result of a sequence experiment. And it's a purely artifactual result that may not even correspond to an actual DNA molecule fragment, as it is a guess, however good of a guess it may be. It might not actually be the real sequence. Therefore, it might not actually be inhering in a real molecule. Likewise, there are some qualities in MSO, such as catalytic ability, that can only characterize the activity of molecules and not information entities, and so they're not applicable to the SO. So in order to solve this, we take advantage of annotation properties in OWL. Annotation properties in OWL are not meant to be logically computable at all. They're just meant to be graph attachments to string literals for human readable purposes and not to enter into computation. But we're sort of getting our way around that by using them as a Boolean annotation properties to filter our classes. So we will annotate entities like SO read with is represented only in SO, and those like MSO catalytic ability is represented only in MSO, so my program can filter these out and understand there's no generic dependence relationship between these and any other counterpart in the other ontology. All right, so here's an example of how SO terms will be automatically generated from MSO terms stored in the master file. Now let's say we have created a term in the MSO called cDNA region, a complementary DNA region, which is a region of a cDNA molecule that is, and it is of course a subclass, It is, of course, a subclass of the broader MSO DNA region. So if cDNA region is not annotated with our is represented only in MSO annotation, we know that the cDNA region in the MSO must be the generic bearer of some SO cDNA region. Okay, we're using identical names now for purposes of convenience, but let's play along here. We can therefore immediately infer that the SO cDNA region must generically depend on, by this relation, on the MSO cDNA. So our program then declares a class for this SO cDNA in our nascent empty SO ontology here. And we don't need to do anything more at this point other than assert that this class exists and it generically depends on its MSO counterpart. And we don't need to assert that it is a subclass of the DNA region, as we will infer that later. So we don't need to copy, say, any subclass of or equivalent to axioms over from the MSO base class. However, if a term, when we go through this process, a term happens to be annotated with is represented only in MSO or is represented only in SO, then we employ a different step in our algorithm. We must copy this class in toto to its respective ontology, including all of their assertion axioms that are required to classify it. So now that we have a whole bunch of SO terms whose only asserted axioms are that they generically depend on some MSO counterpart, we need to have a way to classify the SO to generate its taxonomy. So take our example of the SO cDNA region here. It's milling about around at the top of the hierarchy. That's not particularly classified. It's just a bunch of SO terms here in our nascent ontology. And the one thing we do know is that it's generically dependent on its MSO counterpart here. And since for the MSO annotations, by design, we have a more rich axiomatic assertions in the MSO, we know that the MSO cDNA region is a subclass of DNA region, okay? But since this is, well, it's not flagged as belonging only to the MSO, we have a counterpart in the SO, we know that there has to be a SO DNA region existing somewhere for that generically depends on it. So to classify the SO without having to do any extra work, we just import our MSO with all of its asserted axioms, and we run the reasoner, and voila, we have all of our SO exactly classified using the exact parallel taxonomy of the MSO. So cDNA region is subclass of nucleotide region, is subclass of sequence molecular entity region. And the MSO and MSO taxonomies are kept exactly in parallel, with the curator only have to just adding one term and the program does the rest. So our basic algorithm here, for just to be very, very technical, is we import from our master OWL file the ontology object that it has, and we make copies of it because these are all passed by reference and we don't want to be altering the primary ontology object that we import. Make a copy, say copy A, remove all classes that are represented only in SO true. Well, since we based everything on the MSO anyway, we're done here. The MSO is exported, we're done, we've got MSO.owl. Okay, next we make another copy, say copy B, and do the opposite. We remove all classes represented in only in MSO, and now we're building out the sequence ontology. We have all of our MSO classes, so how are we going to now create our SO classes from it? We employ a little trick. We change our IRIs from the MSO IRIs to our SO IRIs of these remaining classes according to how we wish to do these IRIs. Right now we're just keeping the base 7-digit ID number the same for both of them, but we may change that in the future with a different mapping. Effectively, this declares the existence of all SO classes that are going to depend on the MSO counterparts. We then assert the generically depends on relation to all of them, import the MSO into copy B, do the classification that I mentioned, remove the MSO direct import, and then output this out as SO refactored. And we call it refactored because the sequence ontology has been around for years, but it's been conceptually inconsistent. The taxonomy that we're doing now is completely different from the original version. And it's philosophically more consistent now because we are strictly separating between generically dependent continuants and independent continuants. We have two separate ontologies for each of them. So here we've presented a methodology for designing companion ontologies that other people may have to very closely related ontologies to be semantically and structurally aligned throughout their development life. And you don't need to then just have these two be evolving on their own independently and then try to use complex ontology alignment, what I call post hoc ontology alignment, to try to get them back into sync. Here's our GitHub repository for the code and the project for the molecular sequence ontology. And we welcome contributions from users there, especially those that are using the sequence ontology, have encountered problems with making cross products with other ontologies like the Go that require like molecular entities, independent continuants, and they're finding logical and satisfiability problems with the SO. We really want to bring the SO out of its data silo here. So I'd like to thank Karen Eelbeck, my mentor, Mike Beda, my collaborator. I want to thank Ignazio Palmisano, one of the lead developers in the OWL API. I've never met him in person, but he's like always answering my questions on the OWL API developer list. He's awesome. He's helped me a lot with this. I started out knowing nothing about the OWL API. Some of you may know that it's one of the most gnarly, complicated, headache-inducing APIs in existence, and so he really helped me with that, and of course my funding the National Library of Medicine training grant, T15 training grant. Okay, thank you.
So
sorry for going a little long. Yes.
Yeah, we have time for one question. Please, if you could use the microphone.
Oh dear. So great talk, really exciting work. And I said, oh dear, because this is probably not a short question, but so maybe you could just punt on it. We'll see. So this is great work, and I really encourage and I'm excited about the use of ontologies with OWL where you're actually running inference. And that's where you're going to run into troubles with the BUFO and connecting to things like KEBI or the gene ontology. So my question is really about the motivation and setup for this problem. So do you really have users that are throwing their hands up and saying, oh my God, I can't get inference to work. This is horrible. Aren't there some simpler workarounds where you could just throw away that first equivalence statement, for example, rather than, this is a lot of machinery. Right, so like throw away that equivalence statement and just do it. Would that work? Are there? This seems like a lot of work.
You mean throw away? You mean just say, okay, we're going to just by fiat say that that KEBI mRNA is a generically dependent continuant for these purposes?
Right.
Well, that may be a very nice and sweet solution for this particular problem, but then you've got the propagation problem, where you're going to be going all the way up to generically dependent continuants. You're going to have sisters and siblings off in the distance that are generically dependent continuants that might be involved in relations that are much more complicated than this.
Right,
and, so really
and that's great if they're users that need those.
Yes, and we think there are.
Okay.
Yeah.
We should continue offline.
We wouldn't be doing this if we didn't think that there were users.
So I guess the point that was made yesterday.
I mean, the MSO was demanded by users who have that problem, yeah.
The point yesterday was made that are there killer apps for running inference? And what your talk seems to be implying is, yes, people are banging down the doors that really want inference. And that hasn't been my experience, and I'm happy to hear it.
Okay.
So.
Well, let me know. It may be a domain thing.
Right.
All right, thank you.
Okay, thank you.